Welcome to our seminar on computational neuroscience and AI. Today's lecture is on the preprint, exploring the relationship between brain hemisphere states and frequency bands through deep learning optimization techniques. We've seen a trend in applying deep learning to EEG, with papers like Deep Learning with Convolutional Neural Networks for Decoding and Visualization of EEG Pathology showing its power. This work from researchers at Innopolis and HSE University in Russia pushes into a more nuanced area, not just whether deep learning works, but which specific methods work best. It provides a systematic guide. Yes, Noah?
Hi, Professor. So, is the main contribution the methodological comparison itself rather than a novel classification result? Many papers claim high accuracy on EEG tasks.
That's an excellent point. While they do achieve high accuracy, the core contribution is indeed the systematic comparison. They're addressing a practical gap by testing which optimizers and architectures are most effective for classifying left versus right hemisphere states across different EEG frequency bands. It's about building a reliable and interpretable framework. The central hypothesis driving their work is that the two brain hemispheres interact differently when processing simple versus complex visual information, and that this difference is detectable in specific frequency bands. They used a simple Necker cube and the more complex Mona Lisa as stimuli. Their goal was to see if they could not only classify the hemisphere of origin for the EEG signal, but also understand what the model was looking at. To do this, they set out to evaluate a whole suite of deep learning optimizers, compare 3 different neural network models, a deep dense one, a shallow one, and a CNN, and crucially, use interpretable AI to see inside the model's brain.
Can you clarify the interpretability part? They used SHAP plots. What exactly are they interpreting with those?
Right, SHAP, or Shapley additive explanations, helps them move beyond the black box problem. Instead of just getting a prediction, left hemisphere or right hemisphere, SHAP attributes the outcome to specific input features. In this case, the features are the EEG signal values at different moments in time within a specific frequency band. So, a SHAP plot can show them that, for example, activity in the beta band at 75 milliseconds post-stimulus was a key factor in the model's decision. This connects the algorithm's output back to neurophysiological events.
That makes sense. It's about finding the why behind the prediction.
Exactly. Let's look at their approach. They collected EEG data from 10 healthy participants viewing the 2 stimuli. The raw signals were then filtered into the 5 standard frequency bands: delta, theta, alpha, beta, and gamma. For each band, they trained their models to perform a binary classification task. They tested 3 architectures: a deep 9-layer network, a simple 3-layer network, and a 1D convolutional neural network, which is well suited for time series data like EEG.
Why those 3 specifically? Was the big versus small model comparison meant to test if more complexity actually helped?
Precisely. It's a classic question of model complexity versus efficiency. The deep big model tests the upper limit of what a parameter-heavy architecture can achieve, while the small and CNN models test if leaner architectures can perform just as well, or even better, while being computationally cheaper. What they found was quite interesting. On one hardware setup, the ADAMAX optimizer performed best for lower frequencies like delta and alpha, while RMS-prop was the winner for the higher beta and gamma bands.
Wait, they used 2 different computing systems. Doesn't that make it hard to compare the TensorFlow results from the server with the PyTorch results from the workstation?
It's a valid methodological concern, they acknowledge. Direct comparison between the 2 environments is problematic. However, the findings within each environment are self-contained and valuable. For instance, on the PyTorch setup, the AdaDelta optimizer was dominant, and surprisingly, the small model often achieved the highest scores, reaching a 0.99 ROC AUC in one case. This suggests that for this specific task, a massive network isn't always necessary.
So a simpler model, correctly optimized, can outperform a more complex one.
That's a key takeaway. The beta and gamma bands consistently provided the most useful information for classification, which aligns with their known roles in sensoromotor functions and higher cognitive processing. The SHAP plots for the beta band, for example, highlighted activity around 50 to 150 milliseconds after the stimulus appeared, pointing to a critical window for processing. The significance here is twofold. First, for neuroscience, It provides empirical evidence that hemispheric interactions are modulated by stimulus complexity and are frequency dependent. Second, for AI researchers, it's a practical guide. It demonstrates that optimizer selection for EEG is not a one-size-fits-all problem. This detailed mapping of which optimizers work for which frequency bands is a useful contribution.
And this could have clinical implications, right? The report mentioned that their precision on healthy individuals, around 93 to 95%, is much higher than what's reported in studies on stroke patients. Could this work provide a baseline for detecting pathologies?
That's exactly the long-term potential. By establishing a highly precise baseline for normal hemispheric activity, any deviation from that baseline could potentially serve as an early biomarker for neurological disorders. It creates a foundation for developing more sensitive diagnostic tools. Of course, they acknowledge limitations, primarily the small sample size of 10 participants, so these findings need to be validated in larger, more diverse populations. So to wrap up, this study provides a methodologically rigorous investigation into classifying brain hemisphere states from EEG. It highlights the power of combining optimized deep learning with interpretable AI to generate scientifically meaningful insights. The main takeaway is that for complex biosignals like EEG, the choice of computational tool is highly context-dependent, and finding the right tool requires systematic exploration. But when you do, you can build models that are not only accurate, but also explainable. Thanks for listening. If you have any further questions, ask our AI assistant or drop a comment.